Hello, my name is Mattia Bottom and I will be introducing our project. The issue we have decided to address is hairlessness in the Sphinx cat. We have found that the KRT71 and FGF5 genes are responsible for hair growth in domestic cats. Our goal is to take these two genes from domestic cat and insert them into the Sphinx cat. This should then allow the Sphinx cat to grow hair. Cats with hair have some major advantages compared to the hairlessness cats. Hair provide warmth, so this would allow the Sphinx cat to live in a cooler climate. We will be focusing primarily on this advantage, but there are more advantages I will quickly mention. Hair also is used as a form of communication. Cats often raise their hair when scared or angry. This would allow other animals, as well as humans, get a more accurate reading of their mood. Hair also helps keep cats dry and protects them from external dangers such as cuts and parasites. This would be beneficial because the lack of hair makes them more susceptible to these issues. These are all major advantages that will allow for the Sphinx cat to live in different environments.
Hi, my name is Hannah Gonzalez, and I'll be talking about part of the genetic strategy that will be used in order to achieve our goal in this project. So the first things first, we had to identify the gene that would help the Sphinx cat grow hair. In this case, it would be the FGF5 gene and the KRT71 gene, which would be obtained from domestic cats. And we decided to use domestic cats because they're more closely related to the Sphinx cat. So in order to get these genes, we would have to extract them from the domestic cats by rupturing one of the cells of the organism to let the DNA out. When the DNA has been released, there will be a process of separation of DNA. Since the cell has other materials within, including the genes that we need, when this happens, you will use it as a template in PCR to amplify both FGF5 and KRT71 gene. The gene being extracted needs to be manipulated in order for it to be fully expressed when being inserted into another organism. And our vector would be obtained from an outside company, which would be talked about in a bit.
My name is Nerys, and after isolating the FGF5 and KRT71 genes from a donor, which is a domestic cat, it would then be necessary to prepare the dual sgRNA plasmid vector. The DNA fragments are inserted into a plasmid, which is a circular piece of DNA, and this is done by the restriction enzyme, which acts as a pair of molecular scissors, and it cuts the DNA at specific cut sites, which specifically is going to be the AGEI and the EcoRI. And this then leaves both pieces with sticky ends, which have matching base pairs that are later combined to the vector by DNA ligase. The promoters HU6 and MU6 are also needed to help determine when and where the FGF5 and KRT71 genes are going to be expressed. Since the KRT71 genes are originally mutated in the Sphinx breed, making the Sphinx cats unable to grow hair, it isn't necessary to remove them, and the new genes, the FGF5 and KRT71, can be inserted into the plasmid vector and be sealed into the DNA backbone by DNA ligase at the specific cut sites.
Hello, I am Nico Lau, and I will be talking about some ethical concerns about genetically modifying a domestic cat or animals in general. Of course, there are questions about animal welfare, such as how genetic modification may violate its way of life, and in our case, the Sphinx cat may not be accustomed to having fur. However, it would slowly adapt, and in the long run, the Sphinx would live a more comfortable and warmer life. This is not the first time animals have adapted before. Throughout history, we have seen animals change and evolve to the life that they currently live now. It would not be difficult for a Sphinx cat to be accustomed to hair, especially when it benefits them more than it hurts them. There are also questions about how efficient the technology is, but as of now, technology has advanced to where there are no safety risks against the animal. Concerns such as removing tissue or harming the animal is no longer a problem as it may have been 2 decades ago. As for efficiency, scientists go through multiple processes to ensure that they have the right gene, plasmid, and have went through all the possibilities. Genetic modification has also been blamed to be unnatural, which in our case doesn't hold any real meaning because for the Sphinx cat, it is for a better lifestyle and it does no harm to it. It only changes its ability to grow hair and nothing else.
Hello, my name is Mark Jensen, and for our genetics project, I will be presenting the gene diagrams and the plasmid map. So for our project, we have two genes. We have FGF5 and KRT71. FGF5 is fibroblast growth factor 5. This gene is responsible for long hair growth in certain types of cats. And KRT71, that is keratin 71, is the protein that is actually responsible for the growth of hair. It is actually a mutation in this gene in Sphinx cats that does not allow them to grow hair. So we hope by introducing this gene through a vector, we can have production of hair, even though the gene of the host doesn't really work. At least this way it'll have one working gene. So for our vector, we chose this one. The title's in the center, dual sgRNA HU6MU6. That just means there are two mammalian promoters, one from humans and one from mice. These are shown on the right side of the vector with these arrows, they're highlighted. And after each of the promoters, we have two cut sites. We have EcoR1 and AGE1. We will be using restriction enzymes to cut the vector at these points and DNA ligase to join the sticky ends of each side. The sticky ends of which are indicated here for EcoR1, here's the sequence, and for AGE1, there's the sequence. And you'll see on our diagram back on this side that FGF5 is going to go to EcoR1, and it has the sticky ends just around here. And AGE1 is going to be KRT71, and it has the same. Notice that the sticky ends are after the promoter. Because the promoter is already in the vector, we don't need to add it again. So we will cut it after the promoter, join the sticky ends together to put the genes into the vector, and when it transcribes, it'll start at the promoter, go through the gene and the terminator, and then it will pop off. This mRNA will be modified to receive a 5' cap and a poly A tail. That latter is achieved by this poly A signal at the bottom of the vector. Some other notable things include the origins replication for the vector that is shown by the yellow arrows labeled ORI. That is essential for replication of the vector both within bacteria in the lab and within the host cells. And we also have ampicillin resistance and its respective promoter on the left side of the vector. This is so that when we introduce the vector into bacteria in the lab, we can use antibiotics to kill all the bacteria that did not take in the vector, because the ones that did will have resistance to ampicillin and will survive. So with this, we can add the genes in at the cut sites, introduce that into the cat, and then once it transcribes, it won't have any of these introns, which are indicated up here. The dark boxes are the exons. FGF5 only has 3 and KRT71 has 9. And you'll see from the proteins, FGF5 is actually much smaller than KRT71, which makes sense with the fewer exons. So FGF5 will hopefully make the hair that is produced by KRT71 longer, and ideally the Sphinx cat will not only be redressed in its new clothes, it will have much longer hair, and it'll be available to consumers worldwide in much colder climates, and the hairless one can still be available to much hotter climates. This way the cat can be enjoyed by many more families all around the world. It can enjoy much more environment, and the pet industry will hopefully take this as an improvement.